Hello my dear students, welcome back to Biology at Priyajit. Today we are going to discuss one of the most important topics in microbiology, that is bacterial cell structure and the control of microorganisms. If you are studying microbiology at the undergraduate or postgraduate level, this particular topic is extremely important for you because it forms the foundation for understanding bacterial physiology, pathogenicity, antimicrobial therapy, bacterial taxonomy and antibiotic resistance. So my dear students, in this class we will first understand the important structures of a bacterial cell such as the capsule, pili, flagella, cell wall, plasma membrane, outer membrane, uh, plasmids and bacterial chromosomes. Then we will discuss one of the most fascinating survival structures of bacteria, that is the endospore. Finally, we will move to control of microorganisms including physical and chemical methods, and then discuss chemotherapeutic agents, sulfur drugs, antibiotics, selective toxicity and antibiotic resistance. So let us begin. Introduction to the bacterial cell. First of all, we have to know what exactly a bacteria. Bacteria is basically unicellular prokaryotic organisms. They do not possess a membrane bound organelles, but they also lack membrane bound organelles like mitochondria, endoplasmic reticulum, Golgi bodies, etc. But please do not make the mistake of thinking that a bacteria is a simple or poorly organized cell. Although bacteria have a relatively simple cellular organization, their structures are highly specialized. Even each structure performs a specific function that helps the bacterium to survive, grow, reproduce, adapt, and sometimes cause disease. For example, the capsule can help a bacteria resist phagocytosis, Pili can help bacteria attach to host cells or exchange genetic material, Flagella help bacteria move. The cell wall provides structural strength, the plasma membrane is involved in transport and energy generation, plasmid may carry antibiotic resistant genes. And the bacterial chromosome contains the essential genetic information required for bacterial life. So when we study bacterial cell structure, and we are actually studying the machinery that enables bacteria to survive in different environments, let us now understand these structures one by one. Capsule. Let us begin with the capsule, which is a well organized gelatinous layer located outside the bacterial cell wall. It considered a specialized form of the glycocalyx. Now glycocalyx can occur in different forms. When it is firmly attached and well organized around the bacterial cell, we generally call it a capsule. When it is more loosely organized, it may be referred as a slime layer. The capsule is usually composed mainly of polysaccharides. However, there is an important exception. In Bacillus anthracis, the capsule is composed of poly-D-glutamic acid, which is a peptide rather than a polysaccharide. Because the capsule doesn't readily take up many conventional stains, it may appear as a clear halo surrounding the bacterial cell when special staining techniques are used. Now, why the capsule is important? The answer is protection and survival. One of its most important function is protection against phagocytosis. Imagine a macrophage or neutrophil trying to engulf a bacteria. An encapsulated bacteria can be much more difficult for the phagocytic cell to engulf. Therefore, several pathogenic bacteria capsule formation contributes significantly to virulence. The capsule also helps protect bacteria against desiccation because it can retain water around the bacterial cell. It can also help bacteria adhere to surface and participate in the development of biofilms. Therefore, the capsule is not simply an outer covering. It can contribute to protection against phagocytosis, resistance to drying, adhesion, biofilm formation, environmental survival, and bacterial pathogenicity. Some important encapsulated bacteria include Streptococcus pneumoniae, Klebsiella pneumoniae, Haemophilus influenzae, and Bacillus anthracis. So remember, capsule equals protection, persistence, adhesion, and often increased virulence. Pili and Fimbriae. Now let us move to another important bacterial surface structure, Pili and Fimbriae. These are thin hair-like proteinaceous appendages found particularly on gram-negative bacteria. They are primarily composed of a protein called pilin. The terms Pili and Fimbriae are sometimes used interchangeably, but functionally we can distinguish them. Fimbriae are generally shorter and more numerous. The major function is attachment. On the other hand, some pili are longer and fewer in number and may have specialized functions such as bacterial conjugation. Let us understand their functions. The functions are addition, conjugation, and twitching motility. Now we go for flagella. So the flagella is long, thin helical structure primarily responsible for bacterial motility. It is made up of flagellin protein. It has three major parts: flagellin, filament, hook, and basal body. The bacterial flagella structure is different from the flagella of eukaryotic cells. This is the structure of flagella. So this is the description of filament, hook, and basal body. So the arrangement of flagella: when a bacteria with one flagellum at one pole, it is called monotrichous. When flagella are present at both poles, the arrangement is called amphitrichous. When a tuft of flagella occurs at one or both planes, this is called lophotrichous. And when flagella are distributed over the entire surface, they are peritrichous. Cell wall. The cell wall is made up of peptidoglycan alternating sugar molecules N-acetyl glucosamine and N-acetyl muramic acid. These sugars chains are interconnected by short peptide chain. Now comes a very important comparison: gram positive versus gram negative bacteria. In 1884, Danish microbiologist Hans Christian Gram developed the staining techniques. Bacteria are broadly differentiated into gram positive and gram negative bacteria, whether they retain the positive stain after the decoloration step. The difference is closely related to their cell envelope structure. This is the differences between gram positive and gram negative bacteria. This is the characteristics of gram positive bacteria and cell wall. This is gram negative cell wall. Outer membrane, which is made up of lipopolysaccharide. It is asymmetric structure mainly found in gram negative bacteria. LPS has three major components: lipid A, core polysaccharide, and O antigen. Let us understand them. Lipid A anchors LPS in the outer membrane. It is responsible for much of the endotoxic activity of gram-negative bacteria. When released in significant amount, it can trigger powerful inflammatory responses. This may contribute to fever, hypotension, septic shock, and other manifestations of endotoxemia. Core polysaccharide: the core region connects lipid A with the O antigen. O antigen is a variable polysaccharide that extends outward from the bacterial surface. Its variation contributes to serological diversity among bacterial strains. Porins: the outer membrane also contains protein channel called porins. Porins allow certain small hydrophilic molecules to pass through the outer membrane. However, because the outer membrane restricts the entry of many substances, it can contribute significantly to the intrinsic resistance of gram-negative bacteria to several antimicrobial agents. So when you hear gram-negative outer membrane, immediately think about LPS, lipid A, O antigen, porin, and antimicrobial resistance. So bacterial cell membrane or plasma membrane, it has several selective permeability. It involves in transportation, energy generation, and secretion into the bacterial cell. Biosynthesis: several biosynthetic processes, including components of cell wall synthesis, are associated with the cell membrane or plasma membrane. So never think of the bacterial plasma membrane as merely a boundary. It actually a highly active metabolic interface. Plasmid is the intrinsic genetic element within bacteria, which is a small circular double-stranded DNA that exists independently of the bacterial chromosome. They can replicate independently. Their replication is coordinated with the bacterial cell. Generally, plasmids are not essential for basic bacterial survival under normal conditions, but they can provide important adaptive advantages. So one particular important is the R plasmid. R stands for resistance. R plasmid can carry genes that confer resistance to one or more antimicrobial agent. Some plasmids are conjugative. That means they contain genes that are enable their transfer from one bacteria to another through conjugation. Therefore, plasmids are more contributors to horizontal gene transfer and the rapid dissemination of antimicrobial resistance. Bacterial chromosome: it is basically a single circular double-stranded DNA molecule, also known as nucleoid or genophore. It is not associated with the membrane-bound nucleus. Some bacteria possess linear chromosome, more than one chromosome. Their mechanisms: DNA supercoiling is highly compacted mechanism through DNA supercoiling, DNA binding protein, chromosomal looping, and higher order organization. The chromosome contains genes involved in metabolism, DNA replication, transcription, translation, cell division, cell envelope synthesis, and environmental adaptation. Chromosome versus plasmid. The bacterial chromosome is usually much larger and carries genes essentially for major cellular function. The plasmid is usually much smaller and commonly carries accessory genes that can provide special advantages such as antibiotic resistance or virulence. Chromosomal replication is coordinated with the bacterial cell cycle, whereas plasmids usually replicate independently, and while the chromosome is associated with the nucleoid, plasmid occur in bacterial cytoplasm. So chromosome is the basic cellular life, plasmid is the additional adaptive advantage. Bacterial endospore. Certain bacteria produce highly resistant dormant structures called endospore. Endospore are not reproductive structure. Their primary function is survival, not reproduction. The best known endospore forming bacterial genera are Bacillus and Clostridium. They can survive conditions that would destroy most vegetative bacterial cell. These conditions include high temperature, desiccation, radiation, chemical disinfectant and nutrient deprivation. Structure of an endospore. Exosporium. The exosporium is the outermost layer in many spore. It is relatively thin and contains proteins and no other components. Spore coat. The spore coat is mainly composed of protein. It provides significant resistance to chemicals and enzymes. Cortex. The cortex consists of a largely specialized form of peptidoglycan. It contributes to the dehydrated condition of the spore core. The core contains DNA, ribosome, enzymes, essential cellular component, high concentration of calcium ions and dipicholinic acid. So one of the most important characteristics of the spore core is its low water content. Sporulation. Now, how does an endospore form? The process through which a vegetative bacterial cell develops into an endospore is called sporulation or sporogenesis, which generally occurs when environmental conditions become unfavorable. A common trigger is depletion of essential nutrients. The bacterial DNA is replicated and then a series of developmental changes take place. Eventually, a mature endospore is formed. The mother cell finally undergoes lysis and releases mature spore. Germination. But what happens when favorable conditions return? The process occurs in three broad stages: activation, germination and outgrowth. During germination, the spore loses its resistance properties, take up water and resume metabolic activity. The vegetative cell then grows and divides normally. So sporulation provides vegetative cell to endospore, germination endospore to vegetative cell. Control of microorganism. Now we have discussed bacterial structure. Let us move to the next major topic: control of microorganism. There are different type of agent for the control of microorganism. Firstly, physical agent like heat. Heat is the most widely used method for controlling microorganism, which kills microorganism primarily by protein denaturation, enzyme inactivation, membrane damage and nucleic acid damage. There are two major forms of heat treatment: moist heat and dry heat. So the moist heat is performed by boiling, autoclaving and pasteurization. Dry heat kills the microorganism through oxidative damage and protein denaturation. Filtration. Now filtration works differently. It does not necessarily kill microorganisms. Instead, physically remove microorganisms from liquids or gases. Membranes filters with extremely small pores can remove bacteria from heat sensitive liquids. This is particularly useful for substances that can tolerate heat, such as certain antibiotic solution, enzyme preparation, vitamins and cultural components. Air can also be filtered through HEPA filter. HEPA filters are widely used in laboratory, hospital, clean rooms and biological safety system. So remember, heat kills and filtration removes. Radiation can control microorganisms by damaging DNA and other cellular components. There are two types of radiation: ultraviolet radiation and ionizing radiation. Ultraviolet radiation has poor penetration power, but ionizing radiation has much greater penetration power. It generates reactive species that damage DNA, protein, membrane, etc. Low temperature generally inhibits microbial growth. Desiccation, removal of water from microorganisms. This causes death of the microorganism. Chemical agents for microbial control includes the phenolic compounds, alcohols, halogen like chlorine, iodine, aldehyde, oxidizing agent, quaternary ammonium compounds, etc. There are several chemotherapeutic agents and which have selective toxicity. The drug should damage the microorganism while causing minimal harm to the host. They focus primarily on sulfonamide and antibiotics. Sulfur drugs or sulfonamide, they inhibit dihydrooperative synthase enzyme, reduce folate synthesis, impair DNA synthesis and inhibition of bacterial growth. So sulfonamides are basically bacteriostatic. They control the population of bacteria. Antibiotics that affect cell wall synthesis. They mainly target peptidoglycan. Another important antibiotic is beta-lactam antibiotics. The antibiotic interfere peptidoglycan cross-linking. Antibiotics that affect protein synthesis are such as tetracycline, macrolides, chloramphenicol, aminoglycosides, etc. Amino acids affecting nucleic acid synthesis include fluoroquinolone, rifamycin, etc. Antibiotic affecting cell membrane is the polymyxin B, which target the susceptible gram-negative bacteria. Antibiotic resistance. Antibiotic that has previously affected may no longer work against the bacteria. Bacteria can acquire resistance through mutation, horizontal gene transfer, transformation, transduction, conjugation and other mechanisms. Plasmids are particularly important because the resistance genes can be transferred between bacterial cells. So let us understand the major mechanism of antibiotic resistance. Enzyme mediated drug inactivation. Some bacteria produce enzyme that chemically modify or destroy antimicrobial agent. For example, beta-lactamase can hydrolyze beta-lactam ring of several beta-lactam antibiotics. Therefore, the antibiotic becomes ineffective. Or alteration of target, the bacteria may modify the molecular target of antibiotic. So as a result, the drug can no longer bind effectively. Reduced permeability, changes in membrane structure including poring can reduce the entry of antibiotic into the bacterial cell. This is particularly important in gram-negative bacteria. Efflux pump, some bacteria possess specialized system called efflux pump. So even the antibiotic enters the cell, the bacteria can pump it back out. Metabolic bypass, some bacteria develop alternative metabolic pathways that bypass the pathway blocked by the antibiotic. In this way, the bacteria can continue to grow despite the presence of the drug. Bactericidal agent, the agent that kill bacterial cell. Example include beta-lactam antibiotics, aminoglycosides and fluoroquinolones. Bacteriostatic agent include macrolides, tetracycline and sulfonamide, which inhibit bacterial growth and multiplication. Selective toxicity, finally let us understand a critical concept behind the antimicrobial chemotherapy, that is selective toxicity. For an antimicrobial drug to be useful, it should ideally target a structure or metabolic pathway that is essential to the microorganism but absent or sufficiently different in host. So therefore antibiotics can inhibit bacterial cells while causing comparatively less damage to human cells, and this principle is the foundation of successful antimicrobial chemotherapy. That brings us to the end of the bacterial cell structure and control of microorganism chapter of today, understanding the concept, not just the fact, and connect them with survival, pathogenicity and microbial resistance. Keep learning, keep revising, and subscribe for lucid concept based microbiology and life science lecture for UG and PG students. Thank you for watching. See you in the next class. Bye.